everywhere on the Earth. In the soil, in the water, in hot springs, in radioactive waste, in our bodies, and even deep in Earth's crust. There are currently around 30,000 named species of bacteria, which take a number of different shapes and forms, from spheres to rods to spirals. They were among the first life forms to exist and shape our world in more ways than we even realize. When most people think of bacteria, they think of the awful diseases they can cause. But bacteria are actually much more than just a pathogen. In fact, most bacteria are harmless, and lots of them are even helpful, with different traits that make them suitable for different jobs. Some, like lactobacillus, are used to ferment foods like cheese, sauerkraut, wine, and yogurt. It's even found in our intestines, where it protects us against pathogenic bacteria, which is why it's often used in probiotics. Strains of Clostridium are used for industrial purposes, like in the creation of chemicals such as ethanol and acetone. Others offer an environmentally friendly alternative to pesticides. Others decompose sewage, while others are used to make antibiotics, vaccines, and other medical treatments. Certain biological traits have made bacteria inherently useful to us for eons. And now, scientists are harnessing those traits to rewire bacteria altogether, subverting their biology for our own needs. So what is it about the biology of bacteria that make them so versatile? How has evolution crafted them to be so naturally useful to us? And how are we able to now program them? The beginning of the history of bacteria begins shortly after the beginning of Earth itself. Our planet formed 4.5 billion years ago, and very soon after that, the tree of life started to bloom. Some fossil records point to bacteria's emergence 3.7 billion years ago, with some speculation that they could have emerged as far back as 4.3 billion years ago. All life on Earth began with these single-celled organisms, and over time, as the tree of life branched and diverged, two main categories of life developed: eukaryotes and prokaryotes. The difference is in the structure of their cell. Eukaryotes are typically multicellular with larger, more complicated cells, creatures like the plants, fungi, and the animals. And inside eukaryotic cells, the nucleus and other organelles are enclosed in a membrane. Prokaryotes, on the other hand, consist of just one smaller, simpler cell. Their organelles don't have a membrane. Bacteria fall inside this group. The number of bacteria on Earth is estimated to be 5 million trillion, or 5 times 10 to the 30th power. And these staggering numbers give rise to one of bacteria's superpowers: their ability to rapidly evolve and adapt to their changing environment and food source. In nature, bacteria often grow as colonies, and the populations of these colonies can range from 100 million to 10 billion cells. Bacteria also have a short generation time, the time it takes for them to double in number. It can happen in as little as 20 minutes to 10 hours. And with these two factors combined, evolution for bacteria can happen very quickly. So when they find themselves near a new food source, they often just evolve in a way that lets them eat it. For instance, scientists found that around 350 to 450 million years ago, a group of bacteria acquired a gene that let them break down chitin, a fibrous material found in the exoskeletons of arthropods, which were becoming abundant at that time. While this rapid evolution presents a big danger when it comes to things like antibiotic resistance, these metabolic properties give us a tool to break down things we otherwise could not. Like all living things, bacteria need nutrients to survive. More specifically, they need fixed carbon, or carbon that is incorporated into organic molecules. It's what gives us the energy necessary to move, grow, and reproduce. But it's not as simple as eat food, get energy. Inside our cells, a series of chemical reactions are taking place that break down nutrients into the molecules that our bodies need to grow and thrive. This is known as metabolism, and a really important player in this process are the enzymes that regulate the chemical reactions. In order for a chemical reaction to occur, a certain amount of energy is required, called activation energy. This could be done by adding heat, but that would kill the cell. Instead, enzymes are used as a catalyst. They create a transition state that needs less energy for the chemical reaction to take place. And thanks to their diverse and unique enzymes, bacteria can use almost anything as a food source. While we can only eat organic materials like plants and animals to get our fixed carbon, bacteria can create their own fixed carbon from light and from oxidizing inorganic molecules like carbon dioxide. Having these different options make bacteria much more metabolically diverse than us eukaryotes, meaning they have a lot more options when it comes to nutrients. In fact, scientists have found bacteria eating complex materials that are even harmful to the environment. Several strains of bacteria even have enzymes that can eat hydrocarbons, like those found in oil. After the 2010 BP Deepwater Horizon oil spill in the Gulf of Mexico, resident bacteria ate 200,000 tons of oil and natural gas, aiding the cleanup efforts by converting these harmful chemicals into carbon dioxide and water. But oil is just the beginning of their environmental cleanup abilities. In 2016, researchers in Japan found a colony of bacteria that was eating a common plastic used to make water bottles, known as PET. Each year, around 33 million tons of PET is manufactured. While this only accounts for around 8% of the total annual worldwide plastic production, it is particularly difficult to decompose, even compared to other plastics. This is due to an incredibly strong bond between the two molecules that make up PET, ethylene glycol and terephthalic acid. There were no known organisms able to decompose it until the researchers found a new species of bacteria doing just that. When grown on PET, this bacteria produces two key enzymes. The first breaks down PET into a molecule called MHET. Then, the second enzyme breaks down the MHET into the two components of PET, ethylene glycol and terephthalic acid. The bacteria can then use these molecules as its carbon source. This process doesn't happen instantaneously. It takes about 6 weeks for the bacteria to degrade just a 60 milligram PET film. But in recent years, scientists have succeeded in making a slight change to the structure of the enzymes to make them work 20% better. It now takes only a few days to degrade PET. And with further work optimizing the enzymes, scientists think this plastic-degrading bacteria could be in use, helping us to tackle our plastic pollution problem in just another couple of years. But beyond just altering their enzymes, and beyond their metabolic properties, bacteria hold the key to perhaps the most powerful biological tool ever to exist. And this key is inside their DNA. When you think of DNA, you probably think of the double helix that lives inside our cells. Eukaryotes like us have very complicated DNA. It's linear and coiled tightly inside the nucleus of the cell or sometimes in other organelles, all of which are bound in a membrane. Eukaryotic DNA also contains multiple copies of the genome, many chromosomes, and a large number of genes. But the situation for prokaryotes is much simpler. They don't have a nucleus, and instead, their DNA, which is circular, is stored in their cytoplasm. Prokaryotic DNA also contains just one chromosome, a small number of genes, and only one copy of each gene. This, coupled with a simple DNA structure, makes it relatively easy for scientists to introduce mutations and rewire genes to behave in useful ways. One recent development involves using bacteria for arsenic detection. In regular bacteria, if arsenic is present, an alert protein is released. But by inserting a reporter gene into the bacteria and linking it to bacteria's natural arsenic detection gene, scientists are able to cost-effectively detect arsenic in groundwater. When the alert protein is released, the added reporter enzyme, beta-galactosidase, is also released, lowering the pH of the water, which is an easily measurable change. And now, scientists could even be on their way to curing cancer using programmable bacteria. When cancer takes hold, some of the body's cells begin to divide without stopping. These extra cells may form the growths we know as tumors. In the last decade, treatments for tumors attempt to use the body's immune system to fight it. However, only some tumors respond to this immunotherapy. and the treatment can be hard on the rest of the body. But by programming bacteria to act like a Trojan horse, scientists may have found an answer. It starts with a strain of non-pathogenic E. coli. Researchers added a gene to their DNA that creates what is called an encoded nanobody. Nanobodies are tiny antibodies that bind specifically to a single antigen. In this case, it's a nanobody that binds to the CD47 antigen. The CD47 antigen is one that lives on the outside of cancer cells and tells the body's immune system, "Don't eat me," allowing them to sneak past the body's defense system and continue to replicate. But when the programmed bacteria are injected into the tumor, they begin to replicate in the tumor's core. When their population reaches a critical threshold, they self-destruct, releasing their payload of nanobodies. They bind to the CD47 antigen, and the "don't eat me" signal is turned off, and the tumors become vulnerable to an immune response. Immune cells like macrophages then ingest the tumor cells, and the tumor shrinks, and the healthy tissue is left alone. In mice with lymphoma that were treated using this method, the tumors completely went away, and the mice went on to live normal lives. The researchers are now testing the safety and effectiveness of the method with other types of cancer in mice. After that, they hope to proceed to clinical trials in humans. This is maybe the most promising development in cancer treatment that we have seen in decades. Bacteria have been considered to be our foe for so long, giving us tuberculosis, the bubonic plague, cholera, among the worst things you can imagine. But now, the tables are turning as we begin to understand the huge potential of these microscopic organisms. And as we learn to genetically program them with even more functions, they will soon exist in our world like small organic robots, working to solve some of our most difficult problems. I love learning about science topics and the problems our world faces. But over the last few years, I've been frustrated at myself for not reading as much as I used to. When I was a kid, I would devour books like there was no tomorrow. Why did I stop? It's hard to find the time to just sit still and read when juggling the responsibilities of adult life. Grocery shopping, cooking, cleaning, driving around, commuting to work, exercising, it's all just a lot. But with an Audible membership, you can regain those otherwise boring hours and turn them into time you actually really look forward to. I learned to love my walk to and from work. An hour a day that I resented losing turned into quality time that got my brain engaged and put me in a good mood for the rest of the day. In fact, I even trained myself to love running with Audible. I find a book that I'm super excited about, then only let myself listen to it when I'm running. It's all so much more enjoyable when you can multitask with an audiobook. I shamefully have finished